I think it's time to shift gears. And Perry has the honor to close the presentations today with his very interesting presentation and also lessons learned. I should say that Perry joined 2 years ago, joined ICON 2 years ago, and I'm extremely happy with that and extremely happy that I can work with Perry, given his extensive experience in both pharma and CRO for the past 30 years or so. Perry will cover the phase 1 unit perspective, but will focus more on lessons learned. And Perry, the floor is yours.
Well, Ronald, thanks very much for your very kind introduction. Also from my side, a big pleasure to work together with you. So in that sense, we're now equal. The theme of the presentation, you I think expressed it very clearly, lessons learned after this, so to say, roller coaster, which we all experienced in the last, well, almost 2 years. Lessons learned, interestingly, the main theme of my presentation, I would say, refers a little back to what Ingrid Klinkman in her presentation today posed as a question, and actually she mentioned it twice. And she said, what makes a phase 1 unit safe in the specific COVID situation we are in? As you talked about the biotope we are creating in a phase 1 setting, and of course having the opportunity of intensively monitoring our volunteers, our subjects, but at the same time having to deal with interferences there, for example, staff coming in on a daily basis, which may bring in infection. And it's an interesting and extremely relevant question to answer. And I think all of us working in the sponsored or in the CRO business realize and accept the fact that safety of our volunteers is the most important component of our work. So actually, I will not be able to answer this question of Ingrid completely, but at the same time, I'm really happy to share the experiences and the actions we performed in our unit with a specific focus also on the question of the safety of our volunteers. So, as a short introduction of our site, to give you a reference, here a chart with the information on our ICON CRU, Clinical Research Unit in Groningen. It's a unit, well, if you see the building here, you see that's a new building, it's 5 years old, purpose-built facility, meaning that we designed this building specifically according to our needs, giving us a lot of opportunities to design the wards and the various rooms and laboratories in a way we believe that it functions the best. The basis of our unit is actually already an experience of 35 years. The company started as an independent company, Pharma Bioresearch, which was actually the first commercial phase 1 unit, which was established 35 years ago, was acquired by PRA roughly 10 years ago, and of course since July this year, PRA was acquired by ICON. And this unit is part of a network, as Ronald already mentioned, of 6 clinical research units, which ICON is having globally available. 3 in the US, 1 in Groningen, 1 in Prague, 1 in Budapest. Our unit is the biggest phase 1 unit in Europe. We have 150 beds, including 52 beds, what we call intensive pharmacology unit. And it means that in those 52 beds, we have continuous 24/7 safety monitoring equipment and whatever system available, helping us in providing all necessary safety measurement and monitoring for specific studies where we believe that this is mandatory or helpful. Hospital-based means that our unit is linked to a local hospital in Groningen. It's called the Martini Hospital via an air bridge. And for us, this is extremely important because with this bridge and the contracts we have with the hospital, we have access to the emergency safety support or medical support when that would be required. On one hand, we can ask various specialists when we run into a situation where our own clinical staff needs backup. And in a worst case situation, when acute medical support, resuscitation, or those kind of measures is required, we are linked to the crash system of the hospital, meaning when we push the alarm button, directly the resuscitation team of Martini will come via the air bridge and helps us there where required. And whenever it would be necessary, it's for us very easy to transport someone to the hospital. Of course, those measures, you always hope that you would never need them. It's very rare, but at the same time, it's an important safety requisite. Additionally, we have a fully automated central lab in-house, allowing us, which is 24/7 operational, which gives us the opportunity to directly collect any safety information, safety samples you would require. We have an admin microdose facility allowing us to run 14C and microtracer studies. Surgical room, a little bit comparable, but also Agnieszka mentioned by Biokinetica. We have an in-house GMP pharmacy, which is for a lot of our sponsors extremely helpful because we can produce, so to say, various formulations in a very small scale with very limited stability necessity. We run around 60 phase 1 studies a year. It is quite a substantial number, meaning that we dose more than 2,000 healthy volunteers, either in a single or in a multiple dose setting. And given our facilities, we have the opportunity in principle to conduct any type of clinical pharmacology study, including complex first-in-man studies and all kind of studies you can think of, including biologicals and biosimilars. So in that sense, we position ourselves more, so to say, what we call on the high end of the scope. And of course, when the whole roller coaster of COVID started, it had, of course, some major implications on our business. And I'm happy to share that, of course, with you. Maybe a few things on timelines out of our perspective. I think Ingrid and Stephanie already talked a little bit about timelines. In the Netherlands, it started for us in the beginning of March, where we got more and more positive more and more infections in the Netherlands with COVID. And we started with an order for our colleagues, our staff, to stay at home in case of complaints, which could be related to COVID-19 in the beginning of March. And what we also did is introduce disinfection stations at central places, including also measurement of temperature and a small health check for symptoms for subject and employees of PRA at that time. Those days, of course, things developed. March 13, was an order of our government that we should work from home as much as possible, and we restricted external meetings within our facilities, trying to keep as many people out of our clinic just enough call in order to try to start protecting our biotope, as Ingrid would call it, in our unit. March 16-17 was an important date because then the MHRA and also the EMA came with a first version of a guidance, I would like to call it, where, so to say, a risk for working with for clinical studies in the COVID situation was addressed. And in principle, in those directives, it was mentioned that we, so to say, as a sponsor, but also as a CRO, should take care of potential risk for volunteers or for patients participating in studies. For us, it meant that we went to a discussion, first of all, internally, where we decided that out of a safety perspective, we would like to put all studies on hold, studies that were using investigational products that may have an impact on the immune system. So what we did is, and at that time we had roughly 26 studies in parallel ongoing. So what we did is we evaluated each individual study, did a risk assessment, involved scientists and PIs, of course, in that, simply looking and trying to answer the question, do we see a risk, a potential risk for the participant in this study? Because logically spoken, if you have a compound which has an effect on your immune system, an infection with COVID, of course, could give rise to very serious consequences. Interestingly, of course, we discussed it also with our sponsors. And what you then see was for us quite an interesting phenomena. There were some sponsors who proactively came to us directly saying, okay, based on this guidance which was coming out and based on their own risk assessments, we want you to put studies on hold. So that was roughly 3 or 4 of our sponsors came proactively. At the same time, we also had several sponsors who came to us and said, "Hey, can you do anything to keep the study ongoing?" And of course, various elements always plays a role. Safety is for each sponsor important, but of course, at the other time, strategic considerations, timeline, budgets, they all play a role in this kind of decision making. So what we at the end did, we made this risk assessment, and we at the end agreed with all of the sponsors that we would put those studies on hold, because at the end, that's an important responsibility. We believe that, of course, we have our own responsibility when it's about safety for our volunteers. But sometimes you can run into difficult discussions, of course, with the sponsors. So then it went on. What we did in March 22nd is that we split our clinic into 4 compartments. At that time, and of course it was a time that there was no PCR testing available, we said from, okay, suppose we would have a contamination in the clinic. If we have our clinic in various compartments, and what we did is that we divided the staff into 4 groups which were only working and active on their own compartment. So this was a precaution measure that if we would run into an infection in one of the wards, that we could at least isolate it and may be able to continue with our work in a safe way. Of course, it had some operational consequences, as you can imagine. It takes away some flexibility of your staff, but we felt at the time that it's worthwhile doing it like that. And looking in retrospect, happily, we never needed to make that operational, in the sense that there was a contamination in one of the wards. March 26, we implemented the personal protective equipment in case that social distancing 1.5 meters was not possible, so masks, glasses, and gloves. In the Netherlands, we were relatively late by implementing face masks. In the first weeks of the infection, I think the whole of Europe was wearing face masks, but in the beginning, our government was advised by many scientists, of course, was under the the impression that face masks themselves did not or do not bring much extra safety when it's about contamination, because there was a strong belief that the wrongly handling of face masks, which you of course very often see, touching the face mask, taking it off, putting it on, may even give rise to further contamination. So it was at that time. Nowadays, the situation is different, and the use of face masks is endorsed in the Netherlands like it is in many other European countries. But then on March 30, so that's actually 2.5 weeks after the moment we discontinued part of our studies, which, by the way, at the time was 75% of the studies were cancelled, given this potential influence on the immune system. But on March 30, we got an announcement from the Martini Hospital that, given the rapid way the pandemic was developing in the Netherlands, that they could not guarantee, so to say, ICU support out of the hospital. At that time, most of the intensive care beds were occupied by COVID patients, and we were approaching in what we call in the Netherlands a code black. And the hospital told us, well, in this situation, we can't guarantee that you would have a bed available when something happens. And for us, that was a clear decision point to shut down and stop with all of our work, because we felt that we could not guarantee the safety of our volunteers in the way we wanted. So this was quite a big discussion for us. It meant that on April 5th, the last subject left the clinic. You, of course, always need a couple of days, as Agnieszka also mentioned, that after you stop with dosing, you still have your safety obligations and some safety follow-up before you can safely release a subject in the study. Then on April 8 in the Netherlands, only at that point in time, an official notification from the CCMO, which is our competent authorities in the Netherlands, was published that all phase 1 clinical research should be halted, with the exception of COVID-related research. So at that time, we have in the Netherlands 3 commercial active CRUs, so phase 1 studies. All 3 already were completely closed, but April 8th, this official notification arrived. Then in May, so that is roughly months later, which was for us in a sense a positive moment, the CCMO, in combination with the Dutch Inspectorate, the Health Inspectorate, came, so to say, with a new notification which allows the restart of studies under very strict conditions. This was an interesting moment, because we were in discussion with the CCMO, just talking about on what way we could, on a safe way, start clinical research again. And actually, this is the list of prerequisites which were formulated in order to be able to start. And I won't go, of course, in the full detail, but what you see is that no research with investigational products that affect the immune system. Okay, the social distancing and the personal protection, of course, but one of the elements was that volunteers are not allowed to use public transportation. At that time in the Netherlands, public transportation was only allowed for people who had a need to use it in order to go to their work, and participate in clinical studies was not considered to be allowed. And at that time, we were already planning ways of adapting there and setting up transport ways with buses and so forth in order to bring people in. But luckily, this was not kept very long. An important prerequisite is the fact that volunteers, which would need to stay at least one night in our clinic, need to be tested for COVID-19 before admission. And what we did, similarly to what Biokinetica also did, is that we established at that point in time a set of PCR tests, the Menarini Vita PCR machinery, which is a fast test giving results within an hour. So we in total bought 9 of those machines, so that we had the opportunity on one hand to test all of our volunteers, and at the same time also testing staff, which, so to say, was having complaints or symptoms which could fit with potential COVID infection. So infections of the upper airways, as is basic, so symptoms of the upper airways. That time it was discussed and was ordered that employees should be tested for COVID once a week. Which at that time was quite an issue, what we discussed directly with the CCMO and the inspectorate, because we felt that this was a kind of a pseudo certainty. It is from the logistical point of view, it is quite complex, but you test someone negative who can test positive 3 or 4 days later on positive. Because you can have someone who is just in the conversion phase. So from that point of view, it didn't make that much sense. And also from out of a statistical perspective, if you are going to test a lot of, so to say, people who are not infected, simply given the specificity and the sensitivity of the test, you will always land up with quite some false positive people. So at the end, this was rapidly abandoned again from the authorities. What was another important factor, and that is still in place, is that our CCMO, in conjunction with the Medical Ethical Committee in the Netherlands, responsible for phase 1 studies, requires study-specific amendments for each study, which contains regulation and a risk analysis. And this should, so to say, be submitted to the MATC for review and for, so to say, approval. This risk analysis, and this is comparable to what already was mentioned by Wendy in the presentation, means that we have to assess potential risks with regard to the specific IMP given to our subjects within the study, and assess whether we would see any risk, of course, including a mitigation strategy for those studies. So that's what we also implemented and what we are still doing. So all of our protocols contain a clear assessment and a mitigation strategy. As mentioned, the testing employee was rapidly cancelled, of course, directly leading to a question, and I will come back to that later on. And this was one of the points also Ingrid was mentioning. So what do we do with our staff if we at the same time want to keep our facility and our wards as clean as possible and not getting them infected with COVID? So based on that, we started screening. So we started screening in the middle of June. And we were able to first dose subjects after the closure in the beginning of July. Agnieszka mentioned that recruitment was an interesting challenge in this period of time. For us, it was also interesting, absolutely. We expected a lot of difficulties in that period of time, but we were positively surprised by the interest of our volunteers to participate in studies. And most probably it has to do with the fact that at that time all the schools and universities were closed. We have relatively a lot of students in Groningen and surroundings which often participate in our clinical studies, and well, in a sense, they had time for it. The social life is a lockdown, of course, there weren't that many other opportunities. So we didn't see any problem in recruiting a sufficient number of volunteers for our studies at that time. Of course, we also had people who had some anxiety for participating in studies, specifically somewhat elder population in some studies. They had somewhat more concerns, but this was for us actually not an issue at that time. Then in August 25, we had a further update on the restrictions. So there was quite some relaxation on, and this was of course after the cessation of the first wave. So we didn't had any restrictions with regard to age or underlying conditions with BMI, which is a risk collective in principle, unless of course specific for the study. The RIVM measures, that's so to say the institute in the Netherlands, which is about the advices with regard to the hygiene and all of the measures necessary for containing the COVID in the Netherlands. So all of those measures, the sort of hygiene, social distancing, the face masks, everything is regulated, so they are still in place. There was a clear need for subjects to be tested for COVID upon admission. This risk analysis is already mentioned. And at the end, this was also important. They mentioned that restrictions for clinical research in units should be equal or comparable or equal, they say, to clinical research in healthcare institutions. So they, at that time, said, okay, the same prerequisites we have in the hospitals where our phase 1 studies are being performed now also apply for the phase 1 units. So this was for us, in principle, very useful and helpful situation. First doubling of subjects after closure, just to mention that, you probably would have calculated that it meant for us that we were closed for 3 months. So this is something which, of course, had quite an impact on the operational level, but of course also on a financial point of view. We are a big unit, and of course, this had a massive impact in, so to say, our financial situation. But and we were very lucky with that, and Ronald mentioned that already. We got permission from our headquarters to keep all personnel aboard, and were therefore able to start and restart very efficiently and very quickly. And what is interesting from a planning perspective, when we stopped studies, we stopped 26 studies, we halted them. And of course, we had to restart them all as quickly as possible. And you can imagine when you talk timelines and planning with customers, you can't do everything at the same day. We had quite some interesting discussions with our sponsors on priorities and whether studies could be started more rapidly. So that was not an easy task. It took a lot from our planning department, especially because we, of course, still had a lot of work already planned for this period in time. So we had to deal with halted studies and new studies. Of course, the halted study got the highest priority. So at the end, what we did is trying to squeeze as much together as possible. I think we were successful in that. We never done so much studies in parallel as specifically the few months after the restart. At the same time, reaching the maximum of what really is possible within the facilities. But we were really very, very happy that we could serve our customers in the most optimal way there. Let me push my button, and now my button has disappeared on the system. So. Oh, yeah, here it is. Ongoing measure. Out of all of those measures which you already mentioned, actually since August, September last year, we are still in the same situation. the guidances of the government didn't change. And also from our own perspective, we believe that by doing it the way we are doing it now, we bring in the most possible safety for our subjects. So what do? We have a health check and temperature measurement still in place when entering our clinic building. The hygiene instructions, so disinfection of hands and everything related to that, the sneezing and so forth, and social distancing 1.5 meter is in place in the whole facilities, and this holds true for our staff, but of course also for our volunteers. We have a daily registration of complaints, looking for complaints which would fit with potential COVID infection. So all of our subjects are being asked on a daily basis. We now have FFP2 face mask mandatory inside of the whole of the clinic building, unless when a volunteer is on its bed or when it's in the dining room, and for staff when they are sitting on a desk with 1.5 meters social distancing. All of the rest of the time, everyone has to wear this FFP2 mask. We have this PCR test upon admission to the clinic for subjects, meaning when someone reports in, he will be directly tested on PCR. We have to wait in a separate room, which is outside of the clinic, so to say, until there is a negative test, and only then he's allowed to enter our clinic. What we also have implemented is a PCR test on day +2, or a little bit later, a little bit depending on the risk analysis and the protocol, but a confirmation test a couple of days after the first negative test, just to make sure that you don't miss late converters, so people who just got infected before they had the first PCR test. And I'll show you some data which show that this is quite a relevant way and a relevant test in order to obtain our safety. Currently, our clinic is fully operational. Office staff, we have separate office buildings. We have at the moment max of 25% occupancy, and actually this is already old information. Our government gave a statement last week, again putting us in what we call phase 0, meaning that everyone has to work as much from home as possible. So the office is at the moment in phase 0 and has to work from home as much as possible. It also means that we have limited access within the clinic for external visitors. We had a period in time where external visitors were not allowed. At the moment, given the fact that there is PCR tests and given the fact that most people in the population are vaccinated now, we give limited access with a maximum of 2 external people for monitoring purposes or for audits. Up till this point in time, that all had to be done, so to say, on a virtual way, which is not always easy, but which is possible with all of the modern technology. And what we have, of course, we had that from the beginning. We have an isolation room available for positive tested subjects who were already in the clinic. If someone is in the study and tested positive, we will, of course, directly isolate him or her, do the necessary safety follow-up when required, and try to, of course, bring this subject as quickly as possible out of the clinic, either home or in another address where further follow-up can be given. What we have also as an ongoing measure, I didn't talk about that. Our current situation, our volunteers do not only come from the Netherlands, but we also have around 15% of our subjects in the study are coming from Belgium or coming from Germany. And of course, what we had for a longer period of time is that we have to stop, so to say, their participations in studies. It was given by all of those national different regulations which were there. You were all aware of the color codes which were there, on a certain point in time, the Netherlands were colored red, and then the Germans were not allowed to come in unless when they would go back, they would go into quarantine and so forth. So in that sense, we had to further reduce their participation. The same holds true for Belgium volunteers. If I go, I have to find my switch again. Testing protocol, as mentioned, post-dose day +2. We have that it is in principle advised, but almost all sponsors agree that we do it. Because if they wouldn't want to, then of course it would be more difficult to do that. Good employees will be tested, and this already mentioned. We have this Minarini PCR test available for our purposes. Some data. In total, what we performed within our facilities, so internal and the confirmation tests, which partly were external, we performed 14,087 PCR tests. Positive 123 out of them, so that's 0.9%, where you have staff and subjects. You see the numbers there, 31 positive PCR tests for staff and 92 for subjects, and you see the percentages over there. Of course, when you talk about staff, there is of course bias if you would compare it to the incidence in the population, because people with complaints would come for a test. Well, you see that we say only 2.2% of people with complaints tested positively. For subjects who simply come in principle without complaints, this percentage is lower. If you look at the comparison subject employees over time, you see in blue the total cases. You see that subjects and employees, and that is in a sense what you would expect, that the curves more or less look similar. Then I push the button here. When do we see positive tests? What we see is, in principle, we don't test, so to say, before screening, unless it is required by the sponsor, which is not very often the case. Well, most of the studies the sponsor doesn't require a screening test. But what we do is we test nowadays also volunteers who in the screening examination or in the telephone interview before they come to screening notify that they were positive for COVID. And why do we do it? Because we learned that someone who had COVID may test positive for several months after the infection, and we want to have that available and in fact not enroll them in a study at the screening, and not come into the situation that they come for an admission and then have a positive PCR test. Pre-dose, we found 20 cases of subjects who tested positive, which is 21% of the total positive. And what you see is 38 post-dose, so a day +2 positive PCR. So means that out of a safety perspective, we still had 38 volunteers who tested negative at entry, but tested positive at day +2. For us, the consequence is that those volunteers are taken out of the study, will go directly into isolation, and will be reported to authorities also as positive for COVID. How valid is our situation? What you see here is a comparison to the incidence in the Netherlands. That's the blue line, and the red line is, so to say, our cases in the clinic. And what you see is, and for us it is very reassuring in a sense, is that so the shape of the curves are very much identical, and means that what we test within our own population is, so to say, very similar to the way the incidences are in the population. For us, kind of a confirmation that the validity of our testing method, at least, seems to be okay. There isn't an over or an underrepresentation, which would cause or maybe give you some warning signals. Yeah, some conclusions. Going back to this question of Ingrid, are we safe? What we see in our experience is that the incidence in the clinics follows the incidence of a general population, and as mentioned, we see that as in a sense positive. That we in our measuring methods are not finding or missing maybe some parts of the population. What is important with regard to the safety, and it has to do also with the measurements we have implemented in the clinic, is that we have very few cases of a secondary spreading. And those events were at the very beginning of the pandemic, the time that our subjects within the clinic didn't have to wear face masks. So after when we implemented that and we were very strict on the social distancing situations, all of the positive subjects in our clinic never gave rise to a secondary infection of other member of another subject or member of the staff. What we always do is the moment we have a positive subject on the clinic is that we do so to say research on the context some of us having within our clinic, and we test all of the subjects in which there was what we define and call a risk contact, and we never found someone who was positive after those initial weeks before we had all our safety measures in place. And for us, it is a very important message that the social distancing and the face mask seem to work and seem to protect all of our volunteers and also staff, which is in the clinic. The repeat, as was already mentioned, we believe that it is extremely useful to have this repeat and that there are late conversions who you miss at your first test. All of the subjects who were dosed and tested positive at day 2, there were no AEs reported. So they were taken out of the study. There was, of course, always a follow-up, but for happily spoken, there was never anything which had a negative effect on the safety. What we learned and what we did is we built up a very tight relationship with the local municipal health services, GGG it's called in the Netherlands, which do the further follow-up the moment there is a positive test available. And an inspection of the Dutch Health Inspectorate in September 2020 confirmed the validity and the acceptance of all of our procedures. If we talk, that's my last part. Are we safe? Back to those questions. Never be able to answer that in full detail, but we believe that with all of the measures we are taking and based on the data we see, believe that we are at least in control of what's happening, and we are convinced that with this safety elements we take in place, that we are able to guarantee the safety of our volunteers in a way that it is for us acceptable to perform studies. Staff vaccination, Ronald already mentioned that. That is for sure one of the questions we have to answer. We are at the moment not allowed to ask our staff whether or not they are vaccinated. Currently, at the same time in the government, there is a discussion ongoing and a preparation for a law which may allow employers to ask for that. So that is something we have to further evaluate how we deal with that. So that is a question and something to be follow up. There is another interesting discussion with regards to volunteers, healthy volunteers. At the moment, we do not ask, because we're not allowed, in principle, whether or not they are vaccinated. And we do not, so to say, differentiate on that. We are, of course, in the context of an we are allowed to ask, but at the moment for us, we do not take that as a differentiator. Sponsors themselves at the moment don't have preferences whether or not you would only want to have vaccinated volunteers or not. But of course, given the whole discussions which is now ongoing also in Europe on G3, G2, G1, it is for sure something which will further pop up and needs to be further reflected on how to deal with that. Vaccine studies, we also have experience there, completely different situation. We participated in a couple of studies, one of the big vaccines. At the same time, what you see now, and that's a methodological question, different chapter is how do you deal with new vaccines? We have quite some requests of sponsors asking for placebo-controlled studies. And of course, with 85% of vaccinated population, we can't really do a lot of work over there. But that's something from a methodological point of view. So that means, as my last slide, is that it's a slide which was made a year ago at the time where everyone believed that after the first wave everything was fine and in order, and it says second wave. I think at the moment we can write down fourth wave. You see in the Netherlands, in the whole of Europe, of course, a dramatic increase again of the incidences, putting a lot of pressure on the healthcare system. And at the moment for us it's hard to predict what will happen, specifically also with the planning and the conducting of our phase 1 studies. So that's it. Thanks very much for your attention. It's late on a Friday, and of course I'm also very much happy for any questions you may have.
Thank you, Perry. I think we have now covered all aspects of the COVID pandemic very intensively and very extensively